Hi, Michelle Glass here. Welcome back to another episode in our Digestive System Chapter 24, Lecture 24 series. This episode is all about those accessory organs to the GI tract. These include the pancreas, the liver, and the gallbladder. Now, these are considered accessory because they're not actually part of that whole complicated tube. Instead, they're to the side, but we do see that they have ducts that are connecting them directly to the digestive tract. They are all important in making enzymes to help with the digestion of nutrients, and then we will see that there are hormones also being produced here, especially in the case of the pancreas, that are important in regulating the utilization of those nutrients. So let's get started. Our pancreas, we know, has both endocrine and exocrine functions. Endocrine means, hormone production. We had a whole chapter on that. So that means the chemicals are released into the bloodstream, travel from one tissue to another. Now, we already talked about that group of cells called pancreatic isolates. There were 4 different important cells listed here. I'm just reminding you here of 2 that are involved in the digestive system conversation: alpha cells and beta cells. Alpha cells are releasing glucagon and beta cells are releasing insulin. So make sure you know which cells make glucagon and which cells make insulin. Exocrine, by definition, means that the secretions are released into a duct and then transported exactly where they need to work. So in this case, the pancreas has a pancreatic duct that's going to dump these enzymes directly into the duodenum or duodenum. There are 3 that we've talked about already. We know that pancreatic alpha amylase is involved in the breakdown of polysaccharides. Pancreatic lipase is involved in the breakdown of lipids. And then we talked about pancreatic proteases. And we can also mention here peptidases, which are going to be involved in the breakdown of proteins or peptides if it's a peptidase. So we've listed these other places. In addition, the pancreatic juice will not only have those enzymes, but also waters and ions that are going to help with the neutralization of the super acidic chyme as it's dumped from the stomach into the duodenum. The liver has a whole list of functions. We can summarize them as 3 key functions: metabolic regulation, hematological regulation, and also bile production, which we've mentioned previously. Let's talk about metabolic regulation. The liver is going to be responsible for helping with carbohydrate metabolism. So what we see is if glucose levels are low, the liver has stored glycogen, so glycogen will be broken down to release glucose into the bloodstream. We also see that the liver will do gluconeogenesis, which we've mentioned, which is the production of glucose molecule from other organic molecules. But this can go in the other way too. So we can also see that the liver will store extra glucose in the form of glycogen if we do have an abundance of glucose. We also will see that the liver is involved in lipid metabolism. So it will help with the formation of triglycerides and fatty acids and cholesterol. We also see that the liver is involved in amino acid metabolism. So if there's extra amino acids in the bloodstream, it can take this out of the bloodstream and use it to make lipids or use it to make glucose. And then waste removal. So we've mentioned that ammonia that is produced as a result of breaking down of proteins. The liver is going to help convert that to urea, which can then be removed by the kidney. Other wastes as well, toxins and drugs, can be removed by the liver. Your hepatocytes, your liver cells, have a lot of smooth endoplasmic reticulum, which is helping with the detoxification of drugs. And then we'll see some vitamin storage here as well. So the liver will be storing vitamins A, D, E, K, and B12. The liver also is storing iron. So remember, we had that transport molecule transferrin, which is going to transfer iron from our old red blood cells back to the bone marrow. We can also see that being stored and transferred in the liver. And then connecting in with the removal of drugs, we also have just drug inactivation. So as you're looking at different medicines, you also want to look at how long it takes for the liver to break that down. So that helps you to know whether you need to take it every 4 hours or 6 hours or 8 hours, depending on that inactivation time frame. The liver is also important as a hematological regulation area. It is our largest blood reservoir. So we're going to have here a special type of antigen-presenting cell called a stellate macrophage. So it's doing the job of antigen presenting, which we know very well. We've also talked about the liver as producing most of those plasma proteins. So think in terms of like albumins is important in regulating osmolarity and helping to get fluid pulled back into the venular end of a capillary. We also see that the liver will be important in absorbing and recycling hormones such as epinephrine, norepinephrine, insulin, T3, T4, estrogens, androgens, that's your testosterone, corticosteroids. So that's like corticosteroids, what's the one, aldosterone, that we'll talk about extensively. We've already talked about it for regulation of blood volume. Formation of calcitriol, which we've seen in our conversations in AMP1 as important for our absorption of calcium. So there's a huge role here in the liver and helping to absorb and regulate, recycle hormones. Hematological regulation also includes the removal of antibodies and removal and storage of toxins. So we can see here why liver damage can be so significant for a person's health. And then the final job of the liver is bile production. So remember, bile is being produced by the liver, but stored and concentrated in the gallbladder. The liver is producing bile from cholesterol, and the job of bile is to emulsify your lipids. So emulsification is talking about a great big drop of oil and coating it with bile salts so you can break it up into smaller and smaller drops. This allows those smaller drops to have an increased surface area to have greater contact with the lipase that's being produced by the pancreas, the pancreatic lipase. So remember, your bile is going to have a hydrophilic and a hydrophobic part, so it's helping that lipid to be dispersed in your body fluids a little bit easier. The bile salts, remember, are being stored in the gallbladder, so we need to talk about it as our final accessory structure. The job of the gallbladder is to store and concentrate that bile. It is released through the bile duct directly into the duodenum under the control, remember, of the hormone cholecystokinin, which is abbreviated as CCK. Now, sometimes individuals have to have their gallbladders removed. They are still producing bile. That bile is being made by the liver, and it's still being released into the duodenum. It's just that it's not as concentrated, and the release is not timed with the intake of fatty foods in the same way as it is if you have your gallbladder. So the recommendation is usually don't eat a lot of fat altogether at one time. Eat low-fat meals. Okay, and that is it for now.